Defining the food security challenge in terms that we can address with agricultural research is something that is quite challenging in itself. One of the slides I think that I showed you last year is probably this one, which looks at the rate of productivity gain in some of our major international crops relative to the gains in productivity that would be required to meet food demand out to 2050. And I've had the opportunity to speak a number of places in the world this year, in the last year, including to audiences from which, including authors of the paper from which this data is derived. And they've certainly confirmed to me that this view is still fairly much the perspective they would hold, that in two of the major crops that we're certainly focused on, wheat and rice, productivity gains are probably only running at about half those that we would need to meet future demand. And a sense for us in agricultural research, this is the opportunity. This is the innovation gap that we need to fill in terms of meeting global demand. It seems that at some stage price or other factors will kick in and start to balance this, but there'll be a real need for technology to contribute to delivery in these areas. And we also have, I think, in Queensland, an ongoing issue about potential development of more agriculture in the north. We've had governments at state and federal level that have been a little bit distracted in the last couple of weeks, but there has been, at both levels, a very strong focus on developing agriculture in this country and in Northern Australia in particular, and that puts us really at the centre of delivering the technologies that will facilitate and enable any further development of sustainable agriculture in Northern Australia. And I think I'd like to put also our work in that context. I have had the opportunity to speak in a number of other forums this year. I spoke at the Global Food meeting in Mexico, and there really is, I think, a great opportunity for us to be part of efforts internationally to contribute to developing agriculture right across the tropical world. And places like Mexico, there's a really great interest in collaborating with research here in Queensland, as there are in many other places doing quite interesting agriculture and research. And I know Steve Moore is going to follow up on opportunities in beef genomics in Mexico in the near future. So if we look at what the options are for innovation, I always say there are only two things that you can do. You can change the organism, or you can change the way you manage it in the environment. So we can either breed better plants and animals, or we can work on improving the way we manage them in the farming system. And I think the opportunities are in both of these areas and remain to very much equally in both of these areas. If we look historically at how we've achieved productivity gains in agriculture, they've always been a mix of genetics and environment, and I think that will continue into the future. We have to make balanced investments in those two areas of innovation. I'm going to talk much more about the genetics today, and the advantage of the genetic option is that when we develop an improved plant or animal and put it into the environment, or give it to farmers for production, they have the opportunity of obtaining all of the genetic and all of the science that we build into that organism. In many of the things we do in farming systems, I think the potential for improvement is much more dramatic and much more immediate, but the challenge we have is one of adoption. Will the farmer apply the technology the way we'd imagined it? The advantage of the genetic option is that it's all contained within the organism at the point of adoption. And I'd like to say a little bit about the advances in the technology. And genomics, I probably should define. I think of genomics as the study of all of the genes in the organism, not just the gene-by-gene approach. And I've been involved in a series of volumes that have reviewed progress in this broad area in plants, and I'd use those to illustrate where I think we've come from. If we go back to last century, to 2000 and earlier, we had a lot of technologies that were based around using genetic markers that had been developed over a long period of time that were involving genetic linkage between markers and characteristics of the organism, traits of interest. Things like microsatellites, AFLPs, DARTs, all of these technologies were really not necessarily based on understanding the genetic sequence of the organism. Really from about 2000 on, and this really was the pinnacle of the microsatellite era at that sort of time. From this time on until the mid to late 2000s, there was a huge shift towards using the single unit of genetic variation, the single nucleotide polymorphism, to measure variation, and a huge shift in developing these technologies. But this technology was really limited by the fact that we didn't know the genetic sequence of a lot of organisms. There was a lack of sequence data, a lack of data on which to base these markers. And really, until this point, development of genetic markers for selection was really limited by the technology we had to discover them. But around this time, and I think about 2007 is a turning point, we had the emergence of next-generation sequencing. And that really, of course, from that point on, completely changed the technologies that we have available to us. And certainly by the time of this volume in 2013, everything that's in this volume is related to using next-generation sequencing to discover markers and apply them. And so we left behind completely anonymous markers. In this period of moving to using SNPs more widely, we moved to much more association genetics. Now we've moved to an era in which it's all driven by sequencing. I see that the ultimate convergence of sequencing and genotyping is still not quite there, but we're getting closer all the time, and it clearly is where we're heading. And we now have some projects starting that are really going to just focus on that issue of how sequencing becomes genotyping, particularly for many of the organisms we're working with that have somewhat smaller genomes. This volume that I prepared with Agnello early last year, or published early last year, was really based on the technology of probably the year before, and that really was setting out there how we determine all of the differences between any two individuals at the whole genome level, and that's really where we are with the technology now. Although the protocols we give here in this book early last year would seem very dated by 2015 standards, and so the science is really moving still faster, I think, and so I think we will see that ultimate convergence, and some of those implications I'd like to go through with you in terms of particular applications. The other dichotomy in terms of how we apply technology like this in agriculture is really whether we use it to improve the efficiency of agriculture or whether we use it to change the product that we're producing. Do we produce a better agricultural product or do we produce more of it? It's quantity versus quality. And that's always, again, a choice. There's only two things that you can change: the price of the product or how much you produce. And genomics and all these technologies, genetic technologies, have great potential to contribute to both. At the global level, we need all of the efficiency that we can get. But at the Australian level, I think there's much more focus needs to continue to be put on the quality of the product. Australia is a high-cost producing country, but also we want to sell expensive agricultural products to discerning food consumers because that's where the biggest profit is. Our agriculture will be much more profitable if we can produce higher value products. And so there is a real incentive for us here in Queensland to be using our technology to facilitate production of higher value agricultural products. The other trend that really I think is quite important is to understand what's happening in terms of the marketplace out there, what's happening to consumers. And I've talked about it in the past as the globalisation of diets, but here's a paper that defines it as homogeneity of global food. And so what's happening is that the diets of everyone are becoming more like the diets of everyone else. And this is just a fact of greater communication and greater travel in the world. We all eat foods that have more traditionally been sourced from other places. And at the global level, this is really quite a strong trend. Now, what this means is if you do look at the list of species that might be consumed by an individual in any one year, that list has got slightly longer, and people will say, well, that's great, we're diversifying people's diets, and to a certain small extent that's true. But what we're not really doing is diversifying the demand away from the really major food commodities. And probably an example of that would be where we've seen in the last 50 years an enormous increase in rice in the diets of Australians. Australians are eating much more rice than they did a generation or so ago. If you go to Asia, the trend is to bread. We're seeing a huge growth in consumption of bread in Asia. So what we're seeing there is more wheat consumed in Asia, more rice in Australia. That's not really diversifying ourselves away from those two major commodities. It's just moving them around a little bit. We're still putting enormous pressure on rice and wheat as staple foods. And so if you look at the global diet, we still have an incredible dependence on a small number of species. And these trends are actually making this probably worse because we're all eating, again, increasingly going to be eating the same foods from the same food retailers in different countries. So one of the options we've got is to think about domesticating new crops, and I've been involved in a number of research programs that look at that to see whether we can't make a contribution to diversifying the food base, and it could be seen as something quite fundamental to food security. And there are many opportunities to do that in the Australian flora. We have a tremendous range of crop wild relatives. We've had this philosophy that Australia is imported all its genetic resources for agriculture, and that's true. But if we look out at what we might call the secondary and tertiary gene pools, the more distant relatives of food plants, we find Australia is quite rich. And here in Queensland, we find an enormous number of plants that actually have genes to contribute to our major food crops. It's just that none of them are actually the species we've domesticated, or in most cases that's true. So as the knowledge of the genomes is increasing, we're going to be able to access much more some of Australia's biodiversity to contribute to agriculture in quite meaningful ways. And I've been involved with attempts to domesticate some wild relatives of rice, and the way we're doing that certainly is by analysing the whole genome and comparing those with rice. We can very quickly understand what we would have to change to turn these into domesticated plants. And we've seen the first of those, this so-called alpine rice, moving to be commercialised through a spin-off company that's aiming to develop a whole grain rice alternative that can be grown in much cooler climates. My research group's been involved in looking at the great diversity of rices, and these are many of the species, but really most recently we've focused very much more on the very close relatives of rice that we find here in Queensland. And the large effort now that we have is on understanding these rices. So this is a waterhole near the Cape Development Road north of Laura, where we find a number of species of rice, for example. And these species are very close to cultivated rice, and I was fortunate this year to get an ARC Discovery Project funded with the title Australia, the Centre of Origin and or the Centre of Diversity for Rice. We really can still ask this question about whether, in an evolutionary sense, rice has come out of these populations in North Queensland, which really are potentially very important. So what we're coming to understand is that in North Queensland we have very large populations of wild rice. Here's rice growing in the Mareeba area, I believe, particularly large populations across Cape York. So this year we've really tried to understand the evolutionary position of these rices by looking at them increasingly at the whole genome level. And I think Martis here is trying to assemble a reference genome sequence for a couple of these new as yet undescribed taxa. If we look at these Australian populations here, this is a phylogeny based on the whole chloroplast of what we call the A genome rices. So these are all the species that are interfertile with rice that can be crossed conventionally in rice breeding. And we can see that we have this clade here in South America that sits outside the broader clade, and we have rice being domesticated in Asia. These are the domesticated species here, and we have rice separately domesticated in Africa, Barthii being domesticated to form Glaberrima. But what you can see is that these African and Asian clades from which domesticated rice has arisen have as a sister group the Australian wild relatives, and that there's another African South American clade that sits further away. So this puts these in a very interesting position as being amongst the closest of the wild relatives of domesticated rice, and we do believe they represent a reservoir of genetic diversity that will be really important in expanding rice production to new environments and providing a real source of pest and disease resistance. These northern populations have many classical rice diseases, so here we have a new source of disease resistance for rice in Queensland. Just to show you some of the images, we believe that this species with the more open panicles is quite distinct from the one here that you'll see with the very closed panicles. So they're morphologically quite diverse taxa that we're still trying to understand. Last year we had a trip that explored many of these areas on the eastern side of the Cape. This year, again, in the flowering season, we'll be trying to cover much more of the Cape because there's many areas that we haven't yet explored for rice. I'd just like to show this photo. This is the team on the trip last year standing on this sculpture by a Japanese sculptor in Mariba, and this is meant to be a sculpture to symbolise the importance of wild rice and to recognise Mariba as a centre, a world centre of diversity for rice. There's strong, I think, Japanese cultural links with these populations in Mariba because they see rice in the wild as being something that's really important to conserve. And we really need to understand what's there while we still have the opportunity to conserve some of these resources. We're learning quite a lot, I believe, from studying the genomes of these, these wild species, including where the variation is and where it's been lost in domestication. So here we've got a representation of the 12 chromosomes of rice, and we've highlighted in red those areas where we see no diversity. So if we sequence lots of individuals and compare the genome sequences, we find, for example, this area of about 5 million base pairs here on chromosome 5, in which there's just no variation in domesticated rice. There's another area here, it's about 1 million base pairs of chromosome 1, just no variation at all. So we can see where we've lost diversity, potentially through genetic bottlenecks in domestication, and we can find diversity in these chromosomal regions in the wild rice and be quite deliberate at restoring diversity where we've lost it in domestication in important areas. And quite significantly, this area includes many disease resistance genes for which there's no diversity in domesticated rice, but for which we potentially can find a lot of diversity in the Australian wild material. So genomics allows us to be quite targeted. We can actually target our work to particular genes, particular parts of the chromosome, understanding where it is that we need to look for novel genetic variation. And in rice, we're certainly taking it much further in terms of understanding the implications of eating cereal-based diets high in starch. There's all sorts of concerns, of course, about obesity and diabetes and the increasing incidence of those in populations where people are eating a diet that's higher in carbohydrates, for example. And so we're trying to understand all of the genetics, and in rice now we've been able to examine in the domesticated gene pool all of genetic variation and all of the key genes associated with starch biosynthesis. So instead of looking at individual genes, now we can just look at all of the genes involved in starch biosynthesis in all of the individuals in the population and use that to explain how the genetics of the plant is controlling the properties of the starch, and ultimately understand how those starch properties influence the nutritional value and nutritional impact of the starch in diets, in populations that are changing what they're eating. And this is really something that we're taking to new levels in looking at all of this in the wild populations to understand what wild material might offer us. So we now are beginning to be able to explain how different cereals produce different types of starches at a very fundamental level by doing this at the whole level and across species, and understanding how we could achieve any particular starch content or properties that we might like, any particular nutritional impact in any specific species. There's some recent work that Kai has done from Bob Gilbert's lab, where we've been looking at the chemistry of the starches in African rice. And this has confirmed what we suspected, that the rices domesticated in Africa have quite different starch properties. And what we're now seeing, because of the development of high-yielding rices in Asia, is the traditional Asian rices are being displaced by higher-yielding rices coming out of IRI, and they have really quite a different starch chemistry. We have a very high amylose content in the African domesticated rice, and we can trace that to the differences in the genetics of the wild Rhizobathii from which it was domesticated. We're now looking at all of those genes, and we should be able to understand in the near future how we can achieve in Asian rices the sorts of starch characteristics that we find in Africa. And we may be able to shift the starch profile of rice globally in a direction that's got significant health advantages by going further down this path. So I've been talking about rice, and clearly we have a lot of contributions to make, I believe, to food security from working with rice because of its importance. But probably the other major species is wheat, and our work on wheat at the genome level has really been focused a bit on understanding why we've been successful in the past. We really are struggling to get the sort of gains we need in productivity in wheat, both locally and at the global level, and we really need to understand much more the fundamentals. And so here we've been working at the transcriptome. We've been looking in great detail at all of the genes expressed in the developing wheat seed and trying to understand how they influence the potential to make bread and chapatis. We've got parallel projects on chapatis going on in India and bread here in Queensland from the same varieties, trying to understand why one variety is good for making chapatis and another one for bread. What are the genes that differ, and explain that? And we really have got some great data sets that are informing us. I think I gave the hint last year that we'd identified a novel differentially expressed gene in our prime hard wheats here in Queensland, and for the first time I'm revealing the sequence in a slide because we have a provisional patent in place. This is a very small protein expressed during the mid stages of seed development. There's a bit sort of mid-seed development here. It's the most highly expressed gene in the whole of the seed of many of our premium wheats, and it's just not expressed at all in other varieties. So it's very unusual to find such dramatic genetic differences in any gene, but it is incredibly surprising that the most highly expressed gene in one genotype is not expressed at all in another. And the only association we find is with quality. We believe this small protein, very sulfur rich, probably has a role in stabilising bread volume, and so this may be why all of the premium wheats that have been selected from our program over the years carry this particular allele. And so this now gives us a much more deliberate strategy to try and see whether we can use this gene to more quickly add premium bread quality to some of the very high-yielding wheats that have been discarded in the past because they haven't had satisfactory bread quality. So we can make an indirect contribution to productivity gain by bringing into production high-yielding but otherwise poor quality wheats. And we're really keen at the moment to follow this through with looking at this gene in the background of many wheats internationally with CIMMYT and other agencies. Another thing that I'm not sure we'd quite concluded on last year was that we believe that in rice we can understand hybrid performance for the first time by sequencing the genomes of the parents of our crosses and looking at how far apart they are. In maize, we know the further apart the maize parents are, the more heterosis, more or less. So we can predict the hybrid vigour that we'll get. In rice, we know that there's 20 or 25% yield advantage in a hybrid over a non-hybrid. But we have not been able to predict which crosses will give us heterosis. We don't have heterotic groups in the way that we have them in maize. So we've been trying to understand what the differences are. And here for 3 different crosses, you can see 3 different backgrounds. You can see that what we're seeing, we believe, is that there's an optimum genetic distance. These are the number of SNPs, so the number of differences. So this is 200,000 differences between the 2 individuals. If we go up to 350,000 or we go down to 100,000, it's less performance. We think about 250,000 differences between the 2 genomes is giving us optimum performance. We want to try and validate this in new backgrounds, but what it suggests is that there's an optimum genetic distance that we can actually measure by sequencing the genomes. It does seem to be a quantitative effect over the whole genome, and it's not the further apart the better, that there seems to be an optimum. And that is quite a new result for rice. And particularly in India, where we're doing the field trials for this, we need this gain. Shortfall in rice production is one of the biggest threats to food security over the next couple of decades in India. We need to produce more rice off the same land. Here's a 20% gain we might be able to get if we can get hybrid rice production really working in that environment. I'd just also like to talk about some of the work we're doing on horticultural crops. I know we've got in QAAFI a program on developing high productivity small trees. We're working with a number of the genomes of some of those species, which we hope will in the end make some sort of contribution to those overall efforts. I think I mentioned that we'd done whole genome comparisons of the cherry, the peach and the almond, and that allowed us to discover a number of genes that explain some of the characteristic differences between them, and that whole genome sequencing in these species, because the genomes are small, is relatively easy, and really instead of cloning genes in these species, it really is much easier to sequence the genome of a number of individuals with and without the trait and look for the differences. It's cheaper and faster, and really is a very effective strategy. At the moment, we negotiated a collaboration with groups in India to sequence the mango genome. We're working with a number of agencies in this area. We're aiming to have a genome, ambitiously, to have a reference genome sequence by the middle of the year. With this, we hope to be able to access a lot of the diversity in mango that's found particularly in places like India, and that this will provide a new platform for mango improvement. In macadamia, we've really looked at trying to get a high quality sequence for the main cultivated species, Macadamia integrifolia, but we're also working with all of the wild relatives, and I think this has given us some new clues, again, as to how we can capture diversity. These 2 species here are quite small trees, very small trees, but they are shown to be closely related to integrifolia, and we've found that the genetic difference really is between New South Wales and Queensland. Ternifolia, you may know from north of Brisbane, something you find in the Maleny area, whereas We really didn't have a clear understanding of the relationships between these. Now the genome sequences are taking us a long way, and we should understand how we can capture some of the genetic variation from these other species for Integrifolia as part of this, including the genes that control the small stature of these species. We're working with coffee. We have an ARC-supported project with Green Cauldron Coffee that's allowing us to undertake some fairly extensive work on the coffee genome. And coffee is really quite important in the world because it is considered to be the most high-value agricultural commodity. It's the way we shift money from rich countries to poor countries because we buy coffee. And it really is socially incredibly important in that sense. We last year were part of a consortium to sequence the genome of Robusta coffee, and we published that in Science in September. The big discovery really was that caffeine biosynthesis had been a result of convergent evolution in tea and coffee and in chocolate, that these three species had separately evolved. There are different genes involved in caffeine biosynthesis in these three species. And we're really now trying to extend that much further to understanding a whole lot of other attributes of coffee that are important for quality. Our objective is to produce ultra-high quality coffee for local production. We've joined another consortium that's looking more broadly in coffee. Here's some of the very, very exotic looking species in the genus. We now have over 100 species in the revised coffee genus, extending from North Queensland, and this is our Queensland coffee. This is Darren Crane from the Tropical Herbarium. This is again a plant we find on Cape York. So coffee genus now extends from Australia through Asia, Madagascar, and into all of Africa. And so we've sourced DNA at least from very many of these species, and we have a consortium that really is bringing together an analysis of initially the genomes of 13 of these species, but ultimately to be able to understand the genomes of the whole genus and look at how we can bring new characteristics into coffee. Coffee is one of those species with very little genetic diversity. That's Arabica, where we're attempting a genome sequence this year for Arabica coffee, which is a tetraploid, so this is much harder than sequencing Robusta. But this consortium is now letting us look more widely at all the related species. And the analysis we've done here has shown us the relationships between the coffee species. And we always knew that this is the Queensland species here is in a very different part of the family tree. Here's Arabica coffee here. What's been surprising to everyone is that Robusta is so far away from Arabica. As we know, a Robusta is the male parent of Arabica, but it actually is quite different, and we now believe that it's also quite distinct from many of the others in the genus, and we now have evidence that this in turn has a hybrid origin, and that's a result we've only come to in the last month or two, but I think we've now convinced the taxonomists, and people are now reconsidering where Robusta coffee sits in that relationship. We're also, I think, doing some quite interesting work with Dan O'Connor on peanuts. The peanut genome is really just available to us at the moment, and we're devising a system to sequence all the gene space of peanuts using gene enrichment and to compare the sequences of different genotypes. Dan's objective is to understand the genes that control this process, blanching. To produce high-quality peanuts, we need to be able to turn those into those. It's a genetic characteristic, highly heritable. We're after the genes that control that using a whole genome approach, and we're about to sequence all the genes in many genotypes that are poor performing in this process of blanching and others that are good, so that we can develop a selection for that attribute, which is really critical to the quality of premium peanuts. I'd also like to just say a few words about our energy crop work quickly. We're working, as you're aware, on sugarcane and eucalypts, and our field trials at Gatton of many of these species provide a real opportunity for people to look at some of these species in the field. It's fair to say that the decline in the price of oil has taken a lot of the investment out of the market for biofuels, and BP withdrew quite spectacularly from all its biofuel research investments in the US last month, withdrawing around $1 billion of funding, and a lot of academics are in this space looking for a job at the moment, I think. But what we're looking at is a much longer term agenda, and I do note the oil price is coming back up in the last week or two, so we hope that eventually there'll be real interest here. There is still a security interest, and there is an interest for a number of reasons in being able to continue to go down this path, including concern about greenhouse gases and fossil fuels and the like. We've reviewed recently in this review the broad processes and some of the issues associated with developing crops for these processes, and I think we're in a much stronger position to deliver feedstocks quickly for industry in this space. Sugarcane probably is the world's best opportunity for an energy crop. It already is the most utilised in this regard. And we've got, for some time, had plans to get a really high-quality sequence of the sugarcane genome. That is now progressing at a faster rate, and we do expect that we will get there this year. I know I said that last year, but I hope I'm not saying it next year. We have a considerable number of players internationally in this effort, and the greatest contribution has come through our project with the Joint Genome Institute and the US Department of Energy, contributing to the sequencing of what we think at least will be a monoploid composite genome of sugarcane this year. The sugarcane genome is really impossibly complex for most people that work with normal diploids or even normal polyploid systems, because it's a hybrid with different numbers of chromosomes in each group. A very difficult system to work with. But I think it emphasises that if we actually have a high-quality genome, we'll change the whole business of working with sugarcane. It will become something that is tractable, that we can work with if we can get there in these efforts. In eucalypts, I think Adam's back this morning from visiting the Department of Energy, and I think we're making real progress in having high-quality reference genomes of the species from this area, the spotted gums and those sort of species, which we see as the main opportunities globally, because they're better suited to the tropical and subtropical environments where we can grow these species, we can grow energy crops, than the species of eucalypts that were domesticated in the past from southern Australia. Adam's work's confirmed what we've long suspected, that reticulate evolution in the eucalypts is quite widespread, and we see gene transfer here in the eucalypts, not just between species, but it seems extending out to Angophora and other genera. So here we have reticulate evolution across genus. It really does show that eucalypts have some really unique biology, that we can show relationships between quite divergent species at a common location through apparent low levels of gene transfer that may explain some of the biology of this very large group of plants in Australia. One of the species, and I see Garth's here, so he'll be interested. One of the species I've been trying to deconstruct is Henryi. It's an appropriate name for species I'm trying to argue doesn't really exist. We've shown for a long time these trees I found last year on Sanford Road. This is what they call variegata, and this is Henryi. These two trees are only about 50 metres apart, and my guess is that they're siblings, because the evidence suggests that these are probably one species, and that what the taxonomists use to describe the differences between these species is probably attributed due to a single allele. We can't find anything in the DNA at this stage that differs, but now that we've got whole genome sequences, we may be able to find the single base, there should be one, is genetic, that distinguishes these two species. I don't think they're species, but this will be an interesting final resolution of the taxonomic status of that species. What it is that this apparent species is bigger. The taxonomists say this is a different species from that because there's about 10 different things I can describe. Bigger leaves, bigger fruits, thicker stems, taller trees. They're all size. And it seems to me that that's possibly due to a single gene that determines development, a gene that determines hormones or something that associated with the size, and it doesn't have to be polygenic. But this story will hopefully have a conclusion in the next year or two. When we get, I'd like to just say a little bit about climate and where we're going. We've had a consortium that's been trying to apply genomics to support research on how we adapt agriculture to climate. I've mentioned some of our early work on looking at plants growing across environmental gradients and trying to understand at the whole genome level how those individuals differ in those different environments. This is a transact we did with a very distant rice, the alpine rice I talked about from Melbourne, where we were able to look at which genes differ when plants grow naturally in different environments. And from that, we're trying to understand how we can apply that knowledge in agriculture. On Friday, I'm going to Amsterdam. We're having a conference there that Dave Edwards is actually the instigator of, on adapting agriculture to climate change. And this is a meeting that's had quite strong support. And hopefully from that, we'll continue to see the development of new strategies to use genomics to understand how we can adapt agriculture to climate change. One of the issues that I see is that we have to distinguish between mitigation and adaptation. We're all, I think, focused on adapting agriculture to climate change, but there is a potential for agriculture to be part of mitigating climate change, because agriculture does have an impact on things like greenhouse gas emissions, and by changing our agricultural systems, we can make a positive contribution. And I think genomics, again, has a role in understanding some of the strategies that we've got for mitigation. And a good example here is this analysis where we looked at what impact it would have if you replaced forests with agriculture. And in those red areas, you have a lot of carbon being released in the atmosphere if you replace a forest with agriculture. But in other parts of the world, the agriculture actually traps more carbon. In some of the more temperate areas, agriculture actually leaves you with more standing biomass. So we really need to understand the pluses and minuses and the way agriculture can contribute. We need to understand things like how we can breed rices that release less methane. In other words, getting away from paddy rice, dryland rice. So there are genes there that can help us to really reduce methane emissions, and that's really, from rice, I believe, almost important or more important than methane from animals, which I think we have had quite a focus on here in local research. The big excitement is, I suppose, where the technology is going, and we've been certainly focused on trying to understand the pros and cons of some of the different sequencing platforms. We're now trying to get over the horizon and look at some of the new ones that are likely to be emerging, but the technology continues to move at quite rapid rates, and I think I've been I have to say, I've been very impressed with the outcomes we're getting from using the PacBio platform at TRI, where we're getting single reads that are really very long. I think Marta said she had a 40,000 base pair single read. I think we're getting average reads in the eucalypts over 17,000 base pairs. Reads of this length really changed the game in terms of being able to assemble genomes. Instead of trying to stick together lots of little pieces of DNA of a few hundred base pairs, we now can sequence the genome in a much more meaningful way. So I'm looking over the next few months particularly to see that new chemistry, which has really only been available since December, play out in terms of our ability to quickly capture the sequence of many of these species that we're trying to work with. And we need to think about how we're going to apply all this in agriculture. I think a number of people from QAAFI are going to this meeting late next week in Hyderabad. I'll be there talking about some of the potential applications I see for genomics in crop improvement. The last area to comment on is really the importance I see for information technology and computer science and all of this. As we move down this path, we're all starting to collect much, much more data. I've been working with fairly closely with UQ Research Computing, but I think those relationships will become more and more important as our research becomes much more data intense. And in a sense, this article I wrote with Maurizio on for Trends in Plant Science was really about our conception that really much of the research has moved out of the lab. Much of this research is now done in silico. Much of it's done with people's data from other countries. Is much less tied to physical reality and much more something that happens in computers. So really, that's where all the future lies, is getting, I think, control of the information technology issues associated with all of this research. One of the big initiatives that we're really part of is DivSeek. DivSeek stands for Seeking Diversity. It's an initiative, 70 organisations have joined initially, but it's really has most of the organisations involved in crop improvement and plant genetic resources globally now as partners. It's an attempt to really integrate this into a single informatics system. What we are aiming to do is to sequence all of the genomes of all of the seed in all of the seed banks in every collection in the world and put them all in the public domain in a single accessible through an accessible portal, so we can relate all that data and work with it. It's a very ambitious goal. It's a 7 million genome project, if you like to think of it in those terms. There's 7 million accessions listed for sequencing. This will require enormous global coordination, but it's a great thing for us to aspire to. I'm positioning us to be really, if possible, the Australian node in this international effort, and particularly the node where we capture the novel biodiversity that's found here in Australia and link it into this global effort to provide this effectively massive database for global agriculture. And we really are getting a better understanding. This is the phylogeny of flowering plants from our recent review by working with the genome of how we can move right across the plant kingdom to find the genetic variation that we want for agricultural outcomes. I just want to finish with a few ads. We've got the National Plant Molecular Biology Congress in Brazil coming up, and I'll be chairing the session on food security. We're expecting that to be a really quite important meeting in October. But most importantly, we've got TropAg, our own meeting that we're having here to try and bring the world to Brisbane to talk about how we can meet the productivity challenge of agriculture in the tropics. And I encourage you all to be actively involved in that meeting, because we really will be bringing a lot of quite significant speakers here to Brisbane, a real opportunity to network globally without having to leave home, a really great opportunity. I'd just like to finish by acknowledging the people in my lab that are contributing to the work I'm talking about, the PhD students, Agnello particularly, who runs the lab very well and is a key person in the whole thing. Jason, who's contributed from San Francisco, some of the co-supervisors of the students, and some of the other visiting scientists. Thank you very much.